Go.
Hi, I'm Joe Stein.
I'm Jinwoon Kim, and this is the Photosynthesis Lab for Loyola APES. This lab is done by initially finding the oxygen per gram of water inside. After we do that, we put a plant that produces or goes through the process of photosynthesis. In this case, it's allodium. And after we record the water, we record the plant with the water inside to see how much oxygen is produced. And obviously you see here, for photosynthesis, all the needs that are needed are provided for photosynthesis, like light, water, and so it'll create oxygen.
And then here are graphs and data that is recording. The sun provides the energy for most living organisms, but few organisms can capture the energy and transfer it into other organisms. Animals can warm themselves in the sun, but the energy is not incorporated into their bodies. Only plants, through their chloroplasts, can absorb the energy from the sun, incorporate it into molecules, and store it for other organisms. This process is known as photosynthesis. Photosynthesis is a complex process involving numerous separate reactions, but can be simply represented by the chemical equation 6CO2 plus 6H2O. Add sunlight and you get 6O2 plus C6H12O6.
Alright, good. Photosynthesis takes place in two parts, the light and dark reactions. The Calvin cycle creates glucose by absorbing light through its stomata and into its chlorophyll. The glucose that is created is transferred into ATP in the dark cycles. Plants use the Calvin cycle and ATP molecules to create energy to produce more carbon dioxide and water that leaves the plant.
Our prediction was that the dissolved oxygen in the container of water will increase due to the plants performing photosynthesis and providing more oxygen.
I know.
These are our results. After 15 minutes of the plants in the water, the dissolved oxygen decreased from 3.1 to 2.4 milligrams per liter.
And this is the temperature, the orange one, and you see it's at a constant. And this shows the oxygen dissolved rate, and you see a little dip, and then it stabilizes once again. This is the red one is without the plants, and the blue one is with the plants. The reason why the rate of dissolved oxygen went down may be due to the fact that the plants were either dying or poor quality. Maybe they were starved in the tank that they were originally held in. Or maybe because the lighting is either too weak or too distant from the plants to actually allow it to go through photosynthesis.
We believe that to better this experiment, you could first increase the amount of sunlight hitting the plant, also add more plants, maybe in a larger container, or provide rich soil and not just soak the whole thing in water. Hi, I'm Joe Stein.
And I'm Jinwoon Kim, and this is the Photosynthesis Lab. Yes. Yes. The purpose of this lab was to determine how light and different factors affect the rate of oxygen that is created through photosynthesis.
As you can see, our equipment, we have the light, the plants inside the jar. We have the GLX, which records the data of the dissolved oxygen, and as you can see right here, these are our data. In order for photosynthesis to take place, you need three things: sunlight, water, and carbon dioxide. As you can see, the equation for photosynthesis is 6CO2 plus 6H2O plus sunlight equals 6O2 plus C6H12O6.
Glucose is created through photosynthesis, which is broken up into two parts, the light reactions and the Calvin cycle. In the light reactions, chlorophyll absorbs sunlight and CO2 in order to create the ATP to create glucose in the dark reactions, also known as the Calvin cycle. In the Calvin cycle, CO2 is taken in with the ATP from the light cycle and creates glucose, which is then broken down for energy for the plant. I predict, or we predict, that the amount of oxygen that is created will increase over time.
This blue line represents the amount of dissolved oxygen, and as you can see right here, it decreased and then leveled out over time. And this right here illustrates a steady temperature. I believe that the sunlight was not great enough to penetrate through the glass, or our types of plant were not compatible with water. To make this project better or more useful, I believe that you can use different types of plants and more sunlight.
In conclusion, the plants were supposed to have an increase in oxygen, but instead what happened was, due to certain factors, oxygen was decreased.
Hi, I'm Eddie.
Yeah, you're right there.
I'm Jared.
The purpose of this lab is to record how fast we can get photosynthesis to occur. Photosynthesis is the process in which plants take light energy from the sun and convert it into sugar and energy.
Some equipment for our experiment is we have a light here representing the sun for energy. We have water in a plant, Elodia, which represents the plant that's going to be photosynthesized. We have two wires here, one for recording how much oxygen has been dissolved and one for recording temperature, which goes back here to a GLX, which records the data and sends it to us through this laptop where we can graph the data.
The sun provides energy for most animals, but plants have to make it themselves. Animals can warm themselves, but the energy is not stored in their bodies. Only plants can absorb energy from the sun and use it for other uses later. This process is known as photosynthesis. Basically, photosynthesis is carbon dioxide plus water plus sunlight goes to oxygen gas and sugar. This is needed for plant life. Since animals eat plants, this is necessary for animals too. In order for plants to produce this sugar, three factors must be present: sunlight, carbon dioxide, and water. If one of these factors is missing, then the reaction will not produce sugar, or if one factor is limited, the reaction will stop once that supply is diminished.
Our predictions for this lab are that under a controlled environment, the dissolved oxygen levels will remain constant. Once we add the Elodia, though, it will remain constant for a while and then drop significantly.
As we can see, originally the Elodia affected the levels because it significantly reduced the amount of dissolved oxygen. When we started the stir bar up again, it rose significantly. We think this is due to the fact that the stir bar was moving oxygen from the bottom of the bowl to the top of the bowl, allowing the Elodia to consume it. When that happened, it eventually decreased to a much lower level, much as we predicted. The temperature also began to rise, and we felt this was due to the amount of stirring. The energy obviously heated it up and transferred from work or motion energy into heat energy.
We need to use higher tech equipment to increase our accuracy in this experiment. We need to use fresher water because we'll have a fresh slate, and we'll have more precise control. And we need to reduce the use of rubber bands because it affects the accuracy of our experiment.
We learned in this experiment that when we increase the amount of Elodia or underwater plants into water, we decrease the amount of dissolved oxygen.
Thank you. Have a good day.
Hi, I'm Steven.
I'm Steven.
And I'm Adam. And this is our APES Photosynthesis Lab. So this is how our lab works. We start out with the 2 cups, and in the 1 cup we're going to put, in this cup right here, we're going to put this stuff called Eloded, which is a type of plant that will grow, that takes the oxygen out of the water. And right now what we're doing, we're getting a baseline. So right now we're measuring the oxygen and the temperature of the water without it to see what it's like normally. And then we're going to put it in and see how it works with it in there. So this is our lab, and the purpose of the lab is to find out how much dissolved oxygen will be taken away from the water if we put this little plant in there called Elodia. So right now what we're doing is we are finding a baseline for the water. And right now we have a temperature probe and a dissolved oxygen probe in the water to try to read how much dissolved water is in the water before we put in the Elodia. So we have our two probes which are connected to the GLX, which is connected to the computer, and we have these two graphs right here. This one right here is the bottom one right here is the amount of dissolved oxygen, and the top graph right here is the temperature of the water, and those are both pretty consistent. So that's our baseline. And then we're gonna take some Elodia and we're gonna put it in here, and it's gonna get mixed up with that spinning white magnet, and it's gonna photosynthesize with these lights right here, and it's gonna take dissolved oxygens.
Hello, St. Thomas. Here's some information about photosynthesis. Photosynthesis is a process that converts carbon dioxide into organic compounds, especially sugars, using the energy from the sunlight. In plants, photosynthesis uses CO2 and H2O and releases O2 as its waste product. Photosynthesis begins when UV rays, which is energy, is absorbed by the plant from the sunlight. The plant thus is making its own energy, and the waste product experiences oxygen, which is a necessity for our survival on this planet.
Alright, we predicted that the dissolved oxygen levels for our experiment would go down. As you can see, the temperature for our experiment slightly went up the whole time, linearly, but it did not go up very much. Now, if you look at the dissolved oxygen level, the dissolved oxygen level steadily went down, slightly. The dissolved oxygen went down because there was no carbon in the water, so the photosynthesis did not work.
One thing we could have done in this experiment that would be different, that would maybe help the results, would be to perform this experiment outside. Rather than using synthetic light, the real sunlight may give us true results, and we've been able to conclude this experiment better.